Hello everyone, I am Binod Pokhrel, an early researcher in Agriculture and Forestry University, Chitwan, Nepal. I am thankful to organizers for providing me an opportunity to present my research in 24th Global Immunology, Microbiology and Infectious Diseases Summit 2021. My talk is about environmental factors affecting development of crop diseases. This is the content of my presentation. Let's start with introduction. Pathogens in field experience various environmental effects. Almost all plants require favorable environmental conditions for better growth, development, and subsequent production. A crop disease is the dynamic relationship between host and a pathogen that are intimately related with an environment, mutually influencing and resulting in physiological and morphological deformations. It results from a three-way interaction between susceptible host, virulent pathogen, and favorable environment. When plants susceptible to pathogens are grown in areas, various pathogens are capable of causing diseases, as they can resist wide range of environmental variabilities. About 10% of global food production is lost by the crop diseases, signifying a threat to global food security. Temperature is the most important factor that affects development of crop diseases. Temperature is one of the cardinal factors which influences the occurrence and development of many diseases. Most of the fungal diseases are more severe at lower temperature, and bacterial diseases are more severe at slightly higher temperature than that of fungal diseases. Temperature directly affects the susceptibility of plant hosts to diseases, and it also alters germination of spores, rate of inoculum production, and growth of hyphae. Pentalonia nigronervosa, an aphid which transmits banana bunchitop virus, causing bunchitop of banana, can grow faster at 25 degrees centigrade, which is an equivalent temperature for development of bunchitop. Pre-emergence and post-emergence death of seedlings of cereal crops is mainly due to low temperature, while many seedlings bears lesions and little pre-emergence death occurs at higher temperature. Here we can see that almost all the diseases are prevalent between 16 degrees centigrade to 35 degrees centigrade. Effects of elevated temperature on host pathogen interaction. Here, there is increase in severity due to elevated temperature. So there is direct relationship between temperature and severity. When there is increase in temperature, there is also increase in severity. So there is positive relationship between temperature and severity. Moisture. Moisture is also an important factor that affects plant diseases. Most of the plant diseases are predisposed in condition where there is often soil water deficit. However, most plant diseases are more severe in dry soils and some in wet soils. For example, crop root of cabbage, which is caused by Plasmodium brassicae, is more serious in wet conditions. On the other hand, Streptomyces scabies in potato, Fusarium in cereals, and Euromyces agropyrion on wheat. Also, Sclerotium in onion are most severe in dry soil. Some pathogens show their vigorous activity in intermediate soil moistures. For example, Fusarium solani, which causes pea foot rot, is seen in intermediate soil condition. Pathogens attack and develop in the host plant where there is increases water stress condition and host becomes susceptible due to reduced photosynthesis, which inhibits the production of phytoallergens and reduction in growth of plant without affecting multiplication of pathogens. Likewise, temperature, moisture content of soil also affects the severity of plant diseases. Some fungal pathogens like Phytophthora infestans, Pythium divarianum, and downy mildew fungi are less prominent at lower moisture content, but majority of above ground diseases like leaf rust, powdery mildew, leaf spots require sufficient leaf wetness period for infection. Humidity. Humidity triggers the disease progressing mainly by altering infection process, germination and dissemination of spores. Rain and high humidity triggers the infection of aerial plant tissues by the pathogens. Relatively high atmospheric humidity, which is near more than 85%, is favorable for infection to occur and diseases development. For most of the fungal pathogens, leaf wetness is critical for development of diseases. For example, Pyracularia oryzae, which causes rice blast disease, and Puccinia strophimis or striped rust of wheat, require minimum of 5 hours for disease development and infection to occur. In some cases, high humidity does not directly reduce the crop yield, but its effect is seen in marketability of product. High atmospheric humidity influences resistance to invasion by Botrytis cinerea and Penicillium expansum during development of apple by modifying lenticels. Temperature, light, and moisture often interact with humidity for releasing ascospores in ascomycetes. Atmospheric carbon dioxide concentration. The rate of photosynthesis and crop yield is supposed to increase with elevated carbon dioxide concentration of C3 plants due to accumulation of greater amount of photosynthesis in host plants. Biotrophic fungi like rust promote the development of symptoms. Growth and development of conidia, jump tube, and appreciorium of Coledotrichum is retarded at high carbon dioxide concentration, which is around 700 ppm. The modeling research on elevated CO2 and infection conducted on Scotland showed that the severity of late blight of potato by Phytophthora infestans increased in first half of the one season and decreased in second half of the season. Effects of carbon dioxide concentration on host pathogen interaction have no principles and results. It therefore underscores the necessity to conduct researches. Here is effects of elevated carbon dioxide concentration on interaction between host and pathogen. Light and photoperiod. Light is the most momentous environmental factor for circadian regulation. Various studies have shown that light has direct impact on sporulation of different oomycetes and other fungi, and pathogens virulence has been closely linked to circadian clock of fungal pathogen. Plasmopara viticola, which causes downy mildew of grapevine, immature shape of sporangia was observed in continuous light and has no effect on formation of sporangia and growth of mycelium. Iridospores of Euromyces fascioli germinate spontaneously in both light and darkness, but iridospores of Poccinia gramini have greater affinity to darkness. Low light intensity is best suited for Poccinia graminis to cause rust disease, but high intensity of light, which is about 8,000 lux, is found to be best conducive for Septoria tritici on wheat. However, Stomatal penetration by Puccinia graminis on wheat leaves reduces in darkness. Short photoperiod and low light intensity is conducive for fusarium within tomato. Light intensity and day length during pre-inoculation and post-inoculation of inoculum also affect the severity of diseases development. Soil pH. Soil pH is the important factor which controls the severity of plant disease caused by soil-borne pathogens. Some saprophytic organisms in soil show antagonistic effects to some soil-borne pathogens. Moreover, soil reaction may influence the availability of soil nutrients and affects plant growth and vigor, affecting a change in the microclimate within a crop, indirectly affects infection and disease development. Occurrence of clubroot of crucifers is severe in acidic soil which can be controlled by liming. The spores of Plasmodiphora brassicae are unable to germinate in alkaline soil and can't survive in such modifications. For successful disease development, soil pH is also necessary as above mentioned factors. Streptomyces scabies is more prominent at pH of 5.2 and severity of scab lesions is decreased as 8.5. Fusarium wilt of flax is almost controlled at neutral soil pH, that is 7. If disease has to occur in alkaline soil, there should be high temperature, which is around 23 degrees centigrade, high moisture, about 70%, and high crop load, that is 10 to the power 5 to 10 to the power 7 spores per gram of soil. Soil nutrients. Major nutrient elements nitrogen, phosphorus, and potassium sometimes increase or decrease the susceptibility of diseases. The effects of nutrients on disease development can be subjected to rate of growth of host, effects on vigor of plant, effects on cell wall and tissue, and effects on pathogen through the alteration of soil environment. Susceptibility of plant roots to the pathogen differs due to alteration in plant nutrients. This case occurs in root rot of chrysanthemum, which is caused by Phoma chrysanthemicola. This root rot is found to be suppressed at high level of nitrogen and phosphorus. Microelements also influence disease development, but they are not much concerned as major elements. Some deficiency disorders are whiptail of cauliflower due to molybdenum deficiency, thyroid disease of rice due to zinc deficiency, bluesum in rot of tomato due to calcium deficiency, hollow heart of cauliflower and internal browning of tomato due to boron deficiency, Mars spots of peas due to manganese deficiency. In some extent, minor elements favor pathogen incompetence towards host plants. Potatoes are found resistant towards Alternaria solani and Phytophthora infestans, where nickel and cobalt are in excessive amount. Interaction of multiple environmental factors. Although effects of individual factors are regarded as the foremost importance in the study of interactions, number of factors contribute simultaneously for disease development. Most of the fungal pathogens infect severely in the combined effects of high humidity and warm temperature. Plasmodiphora brassicae develops more club roots in cabbage at the combination of acidic soil, soil moisture of 70 to 80% and temperature of 18 to 25 degrees centigrade. Botrytis cinerea develops prominent gray-brown lesions on grapes at 20 to 25 degrees centigrade temperature and 100% relative humidity, and infection decreases when conditions deviate from optimum. Severe reduction in turnip growth can be observed when combined effects of heat, drought, and turnip mosaic virus infection are subjected than exposure of individual factors. The importance of the interaction of various environmental factors on disease has been commended by a number of researchers, but more intensive work would be profitable in respect to many diseases. Environmental impacts on PAMP triggered immunity, which is indicated by PTI. PTI is the mechanism in which conjugate molecules of pathogens or microbes are recognized by plant plasma membrane localized pattern recognition receptors, which are PRR. Pathogen associated molecular pathogen pattern triggered immunity is concerned with prevention of proliferation of non-pathogenic microorganisms. Virulent microorganisms like bacteria cause infection by causing leaves apoplast to be water soaked. Under high humidity, water soaked condition in apoplast favors non-pathogenic bacteria like Pseudomonas, which induces PAMP triggered immunity. These PTI inducing bacteria develop in host plants like tobacco, Arabidopsis, and bean plants. Impacts on effector triggered immunity (ETI). Effector triggered immunity (ETI) is the genetic method of resistance control mechanism against pathogens. Plant resistance proteins like leucine repeat, which is denoted by LRR, and nucleotide binding domain detect the virulent pathogen inside the plant host. This recognition is similar to hypersensitive response, which is a type of programmed cell death. ETI and cell death are continuously activated by the impact of elevated temperature on ETI. Here are the conclusions of my research. The effect of environmental constraints on host and pathogen has positive, negative or neutral effects on crop disease incidence. Sometimes sole factor is responsible for profuse disease incidence. Fungal diseases were found most prevalent at lower temperature and high humidity than bacterial diseases. Most of the diseases were found to occur in dry soils and some in wet soils due to the fact that poor germination of chlamydospores in dry soil and poor germline growth in wet soil. Here are some references that I have included in my research paper. Thank you.